this is chapter 4 on the cell, and these next few chapters will cover the cell in different amounts of details. So today we're going to talk about cells in general, as well as two main kinds of cells that you should know the differences between and how to compare them. So cells make up your body, and it's the smallest unit of a living organism. And whether you're a single cellular organism or a unicellular organism, which means you only have one cell, or a multicellular organism, cells are the basic unit that function in both of these cases. So example of unicellular organisms would be bacteria or yeast cells, and example or archaea, an example of multicellular organisms or organisms with more than one cell would be humans, plants, dogs, for example. So what size are cells? How big or how small are cells? Cells are on the micrometer scale, and the components that are found inside of the cells are smaller on the nanometer scale, which is 10 to the minus 9. Some cells could be really big actually, and if you've ever had an omelet, you know that the chicken egg is much bigger than a micro scale. It's about that big on a meter scale. And even larger are ostrich eggs, which are some of the largest eggs in the world, and they can weigh even 5 pounds. In your body, you have many different types of cells. Some cells are found in your brain called neuron cells, and they have special shapes. A lot of them have a main region and an extended area called an axon. Cells in your skin are called epithelial cells. They have a lot of times really rigid shapes like squares or cubes or rectangles. Sometimes they're flat and flaky, so they have a different characteristic than, let's say, your blood cells, which are very flexible and round and look like donuts. So in your body, you have different types of cells that have different functions. And so depending on the type of cell, it will look different and behave differently. So how do we see cells? For the most part, many cells, if they're not as big as a chicken egg, which many cells are not, you cannot see them with your naked eye. And this way you'd have to use microscopes. And microscope is a way to say looking at small things. And it's a machine, usually manually operated. Sometimes it could be digitally operated and attached to a computer for viewing, but it helps us to see things on a very small scale. And microscopes work to do two very important things: increase resolution and magnification. So the first thing that microscopes do really is magnify something. And if you want to magnify something, it makes it look bigger. So if something is this small and you look under a microscope, now it would be this big. And that is what magnification does. It increases the size of something compared to its actual size. Resolution is another important aspect of microscopy, which is using microscopes to view smaller objects like cells. And resolution is the process of being able to see clearly between 2 points that are close together. And so the easier it is and the more clear it is to see really small components of a structure, the better the resolution is. So let's say you magnified something and you really can't see very clearly in that picture what's in it. It just looks like a blur. That would be an example of low resolution. But let's say you magnified something and you can see all kinds of smaller structures and they're well defined, and you couldn't see them at all when it was smaller and less magnified. That would be an example of better resolution. For example, real-world application or real-world example. Let's say you are on the top of a building and you're looking down at the street. You would see people and cars and dogs and trees, let's say. Let's say you now magnify this field of view and it looks like it's closer. If you're only able to see similar just little tiny people, little cars, or it looks kind of blurry, that's low resolution. But if when you magnify it, you can see, oh, I think that is Jan, and that is Harry, and Harry's wearing glasses and a red shirt today with a zipper on the back. That would mean you have high resolution. You're able to see details when you magnified it. Okay. Now we're going to talk about also one other thing. So you can use a microscope, and there are two types of microscopes, or multiple types of microscopes really. The most common one that you would use, especially in lab, is the light microscope, and a diagram of that would be in your PowerPoint. But the light microscope works with two lenses: an ocular lens, which is the lens you look through, and then an objective lens, which is used to magnify whatever it is you're looking at. And the microscope shines a light through whatever you're looking at. So your tissue or your cells have to be really thin so that the light can pass through, and then you can view it on your microscope stage. And so review what the light microscope is and look at the different parts and learn that. And then there are other types of microscopes that use different approaches, such as the scanning electron microscope or the transmission electron microscope. And these types of microscope uses electron density, or the density of the different parts of the cell, in order to create an image. And it kind of uses ionization with those electron charges in order to create this image. And so the basic thing that you need to understand is that images are created that are much more clear and magnified than you would be able to see with your naked eye, as well as the electron microscope could make images that are at a much higher resolution, so you're able to see more clearly small structures using the electron microscopes than you can with a light microscope. So those are some quick overview of some of the microscopes. You can also study cells in its parts. So I'm using the process known as cell fractionation. This is a process of basically breaking up cells, and instead of having one full structure of a cell or multiple cells in a tissue, you would kind of break it up with, it could be a mechanical way to break it up, or you can use detergents, and that would break up the cell membrane, and the different parts of the cell will kind of come out in a solution. And then you can use this machine known as a centrifuge. And what a centrifuge does is it spins, it has the central axis, and it spins around that axis really fast. Think of like a merry-go-round, or one of those fair rides that you're spinning in a circle like this. And as you spin, if you remember in those rides, if you've been on one, you'll feel this force pushing against you. Okay, that's called centrifugal force. And that force can separate different molecules based on their size. So if you're really heavy, you're going to go further away from that central point of rotation, further out. And if you're light, you stay closer to the central part of rotation. And in this way, you can do what's known as cell fractionation, where depending on how fast you spin and for how long, you can start to separate big items in the cell from small items in the cell. And then you can study the cell components that way too. So these are just different approaches that scientists use to look at and study cells. Now, most of this chapter is talking about two main types of cells: prokaryotic cells and eukaryotic cells. And the goal of this part of the chapter is so that you will have a better understanding of the similarities and differences between these two big categories of cells. And so we'll start with the prokaryotic cells, and then of course I want you to compare and contrast to the eukaryotic cells. Okay, So with the prokaryotic cells, these cells are found mostly in the bacteria and archaea type of organisms. So most of them are considered prokaryotes. And they have a special kind of characteristics that are seen commonly, even though they're different types of archaea organisms and bacteria organisms. And so they tend to be quite small in size, much smaller than the eukaryotic cells, which we'll talk about soon. And they have DNA. So all cells have DNA. That's one thing to know that we know of. Okay, all cells have DNA that we know of. And cells have a cell membrane, which holds the cell together. Okay, so they have a membrane around the cell. Okay. And they have DNA. The DNA is located in a region called the nucleoid. And you might say to yourself, that sounds similar to the word nucleus, if you know that one. But there are two different types of structures. DNA is found in both of those structures or regions, but in prokaryotes, the nucleoid is just an area, okay? So this brings me to the next characteristics. The prokaryotic cells have no organelles. And organelles, if you look over here, these are all of these little different types of structures that are enclosed by membranes. Prokaryotic cells do not have any of these membrane enclosed structures, so they have no organelles. And they have some ribosomes and storage granules to help with producing proteins. They also have sometimes, not always, flagella or cilia that helps them to move around. Because these are unicellular organisms, they don't have legs, so they have to somehow get around. So sometimes they have a flagella or cilia, and that has like a motion action to it. Prokaryotes sometimes also have cell walls, and cell wall is an added layer of protection around their cell. It's harder. Sometimes they even have an additional layer called a capsule that helps to protect them because their whole life is just one cell. So what I want you to understand with the prokaryotes is that they're very simple cells. They have DNA. The DNA is not in any special compartment. It's just in a region called a nucleoid. And they have this region out here called the cytoplasm, you can call it that. But then they have a cell membrane and a cell wall and sometimes flagellum and cilia to get around. And this is a type of cells for bacteria and archaea. All right. Okay, so now we're going to spend some time talking about eukaryotes. And eukaryotic cells are much more complex than prokaryotic cells. There's a lot of details to understand for eukaryotic cells. And I will pause here just to say that understanding this diagram as well as any expansion of it is a really good start to understanding much of what we'll learn this semester. So let's get started. Eukaryotic cells. These are found usually in multicellular organisms like ourselves, humans, and plants and fungi and these types of organisms. Basically anything that's not bacteria or archaea would normally have eukaryotic cells. And these cells have some special characteristics. So they're much bigger than prokaryotes. I tried to draw an example here. The egg cells, the ostrich egg cells, those are also eukaryotic cells. Extreme examples, but they're also very big. These cells also have DNA, so all cells have DNA, but this DNA is found in a nucleus. So this is the nucleus structure right here. You also see all of these different compartments. Compartment. Okay, these compartments are called organelles, that word, organelles. So organelles are found within the cell of eukaryotic cells, and that helps to separate different regions of the cell on the inside. Okay, so each of these organelles have different roles, as we'll talk about. So around the cell, you have a plasma membrane. And we'll get into the membrane more in the next chapter. But this plasma membrane is covering all the way around the cell that protects it from the external environment. And it allows certain items and certain molecules to get into the cell and certain ones to leave. So this plasma membrane is called a semi-permeable membrane. And that means that some things can get in and some things can get out, but not everything. And you want to think that's important because can you imagine if everything in the cell just spilled out, or if random bacteria and toxins just jumped into your cell? That's bad too. So your cell has a way to allow certain things in and certain things out. All right, so inside the cell, which is what we'll focus on today, we will start here in the nucleus. So the nucleus is the organelle in eukaryotes that contain your DNA. And so we talked about DNA a little bit. This is your genetic material. It is organized into chromosomes. You have 46 chromosomes in your cells. And that is all organized and found right here in the nucleus. Okay, so this is your nucleus. There. So in your nucleus, you also have a smaller sub area, it's a small little area known as the nucleolus. And this region also has DNA, but the DNA there serves a special purpose. So your DNA in your nucleus is important to produce certain molecules that your cell needs for information purposes. And the name of that molecule is RNA. So what your cell produces, your DNA, through the process of transcription, it makes this RNA molecule based on the type of DNA that you have in your nucleus. There are different types of RNA molecules. One that we'll talk about a lot is known as messenger RNA molecules. And that RNA molecule is important to make proteins. And we'll talk about that soon. But in the nucleolus, there's a special type of RNA that is produced called rRNA, or ribosomal RNA. And ribosomal RNA is specifically important for the structure of ribosomes. So if you remember, we talked about different organic compounds in chapter 3, I'm sorry. And these, one of them was proteins. And proteins is important for structure. And proteins, you should know, in the cell are produced by ribosomes. Ribosomes are the structures where the production of these proteins occur. So when you join those amino acids together to make the polypeptide, that occurs at the ribosome. But how do you make the ribosome if the ribosome is needed for protein production? So the ribosome is made up of ribosomal RNA. So this ribosomal RNA comes out here, folds up into the structure, and associates with some other proteins too, but that's what helps to form your ribosome, okay? So this structure specifically is formed from the nucleolus, which produces the ribosomal RNA. Now, in order to make your proteins, first you have to get a message from the nucleus. And that message comes from your DNA in the form of messenger RNA. So this is another kind of RNA, but this one is called mRNA. And messenger RNA, you can think of this one as kind of, if you go to the library, there are lots of books there. Just like your nucleus have lots of DNA. But then if you want to use some of that information, you're not going to write down everything in the entire library. No, you're going to take a book and maybe write down a couple of things, or take a copy of something with a machine or with your phone, take a picture, and then you're going to leave with that information. That information you leave with is like an RNA molecule. You take a copy of the DNA and then you take that information out of the nucleus to use it somewhere else. So when your cell makes some messenger RNA from DNA, the messenger RNA leaves the nucleus and it leaves through an opening called the nuclear pore. Okay, so the nuclear pore, this is the space here, and it goes into this ribosomes. Okay, so when the messenger RNA goes to the ribosomes, what is the role of the ribosome? To make proteins. So the messenger RNA has the code that is important to tell the ribosome how to make a special type of protein. So if you want to make a protein that is important for, let's say, the Golgi, it will look different than the protein that's important for the mitochondria. And so those would have different type of messages, messenger RNA, that goes to the ribosome to produce different types of proteins. Think of it as a blueprint or a recipe for your protein. That's basically a lot of the information that you would need to know about your nucleus. It has DNA in it. The DNA gets transcribed into messenger RNA or ribosomal RNA, which are two examples. We'll talk about some others later. Ribosomal RNA is important to make the structure of the ribosomes. Messenger RNA leaves through this pore, nuclear pore, and goes out here to the ribosomes and code for the proteins that you need for your cells in your body to work. Yes. Now, outside of the nucleus, this is one organelle. There's a lot going on in the nucleus. This is what controls all of everything out here. So now we have this structure, the ER, okay? And the ER stands for endoplasmic reticulum. And so the endoplasmic reticulum, there are two regions to it based on what is produced there. So one is the RER, or rough ER. And in the rough ER, what do you see here? These are ribosomes. So on the rough ER, you have ribosomes attached, and this is where proteins are produced. On the other side is the smooth ER, or SER. And the smooth ER, this is where your lipids are produced. All right, so now moving on to other types of organelles that you have in your cell. Here we have what's known as the Golgi apparatus. And the Golgi apparatus is a stack of 3 membranes that is important for packaging, sorting, and modifying proteins. So when proteins are produced by ribosomes on the rough ER, the RER, they are transported over to the Golgi, and they move through the Golgi complex or the Golgi apparatus, and they get modified. So think about your proteins just getting some of those functional groups added, or maybe parts of them are broken off, or maybe they're joined with other proteins or things of that nature. So a lot of modification goes on here, and during the Golgi is where your proteins are targeted to specific regions of the cell. So let's say that a protein passes through the Golgi and now it needs to leave the cell, or it needs to go to the mitochondria or to a lysosome or back to the nucleus. The Golgi would direct where that protein ends up. Okay, so some other organelles that you'll find in your cell, one of them is the mitochondria. And the mitochondria is important for metabolism and specifically cellular respiration reactions, which is important to produce ATP, which is a form of energy that your cell and body uses for many different reactions and functions. And so the process of cellular respiration, which we'll talk about, is an aerobic process, meaning that it requires oxygen. And it requires oxygen to produce ATP. And the molecule that's broken down in order to produce this ATP is oftentimes glucose. So when we eat our food and we break down our food into smaller components, A lot of times the glucose sugar, when it's present, it is used in the mitochondria for the process of producing ATP. Another similar organelle found in plant cells, not animal cells, would be chloroplasts. And chloroplasts are important for producing food, specifically glucose, and different types of sugars for plants. And these organelles would use light energy in order to produce food. And these are some of the two organelles we'll talk a lot about in future chapters as well. And then in your cell, in the eukaryotic cell, you also have peroxisomes and lysosomes. And these are two small organelles that are involved. They have different enzymes that help to break down and recycle different components in your cells, and sometimes food that enters the cell. So they're kind of a breakdown, recycling type of organelle. Now, outside of all of these organelles, there's a space here. And this space is made up of a jelly-like substance called the cytosol. And this area is generally referred to as the cytoplasm, all the area outside of the nucleus and around these organelles. And this is kind of like if you have a room full of different furniture, the space, the air space would be the cytosol, but it's not air, it's liquid. It has water and other molecules in it. Also found in the cytosol are basically some cytoskeletal proteins. So I drew in green to represent that. And these give structure and shape to your cell. And this is not something you really find in prokaryotic cells either. And so you have three main types of polymer molecules that are important for the cytoskeleton. And think about the skeleton for your body, your bone structure, right? It helps you to stand upright and not fall over. So for your cell, it gives your cell specific shape as well. And these are three types of molecules. The largest molecule is called microtubules. And so they're really big, hollow tube type of structure, and they extend all over the cell. And then you have a medium-sized structure called intermediate filament, and a tiny one called actin or microfilaments. And so there's three different sizes. Some of them help with the structure around the cell membrane. Some of them help with the overall cell structure. And microtubules and specifically helps with transport in the cell. And now this brings me to the one thing I want to talk about here for your cell in general. So in your cell, you have all of these different compartments, and they have different roles and different functions. And the functions, as you're hopefully seeing, are not isolated from each other. They're connected. And so now I want to talk about how do different molecules move from one compartment to the next. Specifically, a special type of transport system that moves different items from one region to the next. And that system is called the endomembrane system. And the endomembrane system is a way that your cell moves molecules or proteins from inside one membrane to inside another membrane, and possibly eventually even to outside of the cell. All of this occurs without interacting with the cytoplasm. So let me give you an example here. So let's say a protein is produced, and that protein, you want it to end up outside of the cell there. So if a protein is going to be produced, it has to first start from what is the message to produce this protein. So where would you find this message? You would find it in the nucleus. So the DNA would have the message to make the protein. But the DNA stays in the nucleus, and in order to get that message, you have to make a messenger RNA. Messenger RNA would then leave the nucleus through the nuclear pore, which is part of the nuclear envelope. And that would now travel, the messenger RNA would travel out to the ribosomes, and the ribosomes are found on the rough ER. Now, when your protein is produced, and your proteins, these are made up of amino acid monomers joined together to make a polypeptide. That polypeptide would go inside the rough ER. So your polypeptide would end up in here, okay? Right there. So inside of this ER is inside of the ER organelle. Inside. So now this is not the cytoplasm, this is inside the space of the rough ER. And now you're inside of the endomembrane system officially. So now you have a protein inside the ER. We said proteins can get modified and packaged at the Golgi. So in order for your protein to leave the ER and go over to the Golgi, it has to travel there. And so two things to travel. One, these cytoskeletal proteins, they're found all over your cell, running in all kinds of directions, okay? And they're used kind of as a road or a pathway for things to get from one place to the next. And in order for your protein to get from the ER to the Golgi, it has to travel in a little ball known as a vesicle. So the vesicle is part of the membrane here that would basically be pulled out of the main membrane and form into a little ball of its own. So a vesicle would leave here, would turn into a little ball, and that vesicle would travel along the microtubule there. And not only would it travel, it would also make sure that it has your protein in it, okay? So it would possibly have your protein there if you want your protein to be part of the endomembrane system. So let's review. You made a protein on the ribosome. It ended up inside the ER. The ER made a vesicle, so part of the membrane came off and formed a vesicle. It has your protein, and then this vesicle is going to go and travel down to the Golgi, and it will fuse with the Golgi. Now, once your protein is in the Golgi here, it will get passed through each part of the Golgi, and while it's getting passed through from one face of the Golgi to the next, it will be modified. Means you might add some groups to it, phosphate groups, sulfur hydryl groups, different types of groups, depending on the protein. And once it leaves the Golgi, it will still be in a vesicle again, okay? So always inside of a membrane, okay? This is where your proteins are. And moving from one compartment to the next via vesicles, this is the endomembrane system, never interacting with the cytoplasm. And this vesicle here can go, This vesicle can go and fuse. Let's say if it was something to go to the mitochondria, it could possibly go to the mitochondria and fuse with the mitochondria and deliver the protein, or it can possibly go to the cell membrane and fuse and deliver the protein outside of the cell, okay? So these are how proteins that are made in the rough ER get sent inside of the ER lumen, and then it is transported via vesicles to different parts of the cells, including outside. So that is all called the endomembrane system, how proteins and different molecules get passed from one compartment of the cell to outside or to other compartments without interacting with the cytoplasm. All right, so that is pretty much it for this lecture. I want to say that there are nice videos that can help you to visualize this in a more animated format if this lecture was not enough for you. And another way to study this is to try to create your own version of the cell, your prokaryotic cell and your eukaryotic cell. I would suggest to get a really big piece of paper, or maybe if you're really into computers, do it on a computer. and just piece it together. And as you're saying, okay, let me say this is the plasma membrane. It's made up of these phospholipids, which we'll talk a lot about in the next chapter. And then this is the nuclear envelope, which makes up the membrane for the nucleus. Go step by step and build your own cell. And this will help you to know each part and what the role and function is. And then at the end, go through and say how everything's connected and explain your endomembrane system. Okay, so all of this is a way to study the cell. Yes, the pictures are already made for you, but if you create them yourself and talk about it, is a better way to really grasp and remember what's the different parts of the cell, what those different parts are. Okay, so hopefully this helps you, and of course, always ask me questions if you have them.